Hi, Maya and Ronen. In the previous segments, we talked about how genomic data revolutionized biomedicine. New and constantly improving technologies allow for high-throughput analysis of genomes. This is why there is so much genomic data available and why these data get so much attention. However, there are many more types of data that increasingly become available to researchers and to physicians. The proteome, namely the sum of all proteins in the cell or in an organism, is one such resource. Just like genomics is the science of analyzing the genome, proteomics is the science of analyzing proteins. Proteins are the nanomachines that execute life and are the final products of coding genes. High-throughput technologies, for example, ones that rely on an arsenal of antibodies that specifically bind to different proteins, can measure the abundance of specific proteins in a cell or a tissue under specific conditions. They can also measure the abundance of proteins under specific clinical states. Researchers found that analyzing the amount of multiple proteins at once can teach us a lot about the patient. For example, many studies have attempted to analyze the genome of patients with major depression in order to identify a set of mutations that will allow us to predict or diagnose the disease or to shed light on the molecular mechanisms that underlie depression. But most of these studies have failed. However, proteomic studies that analyze the abundance of specific proteins rather than analyzing a set of genomic variations were able to identify significant differences between patients with depression and healthy individuals, and even develop a diagnostic kit for depression based on protein abundance. Another source of data are what we can find in electronic medical records, or EMRs. The truth is that electronic medical records have been developed not for research purposes, but rather for following up on the clinical care, for facilitating medical administration, and, if you follow the money, for billing. However, it became clear that once these data are collected, they are a huge and mostly untapped resource of medical information. Because of the size, variety, and complexity of these data, they cannot be analyzed effectively without sophisticated computational tools from the field of data mining and big data analysis. It is amazing what can be computationally extracted from these data. One study, for example, has shown that computational analysis of medical records of children, exploring, for example, patterns of admission and hospitalization, may identify suspected cases of child abuse years before human caregivers identify them. Another example, analysis of changes over time in routine blood work led to identifications of patients with very high risk for having colon cancer, even in cases where the results of each individual test is within the norm. These are just two and pretty random examples for the almost infinite potential for breakthroughs that can be obtained from applying data analysis technologies to already available medical and molecular data. Remember that there are many new resources of medical relevant data: molecular, clinical, environmental, and epidemiological. Each type of data can potentially be unlocked to improve our understanding of human health and to allow for better treatment. But each type of data also poses unique set of challenges, both in collecting the data and in analyzing them. The ultimate vision, obviously, is to be able to combine the data from all these sources in a synergistic manner. I hope you get a sense of what we mean when we say that medicine is rapidly changing to become data-driven. It is not easy to select what should and can be covered within an introductory short online course. The idea of this course is not to convert physicians who watch it into bioinformaticians, but we hope to acquaint our viewers with the major concepts, terms, tools, and databases in the field, allowing them to understand new studies and to think of ways to use this science in their own work. We will start with sequence alignment. Then we will talk about the storage and maintenance of biological sequences in databases. We will introduce BLAST, the search engine for sequence information, and explain how to use it. Then we will move to describe massive parallel sequencing, which is often referred to as next generation sequencing, even though it has been around for a few years already. This is the technology that enables us to sequence human genome and other genomes. We will discuss its application. Then we will talk about electronic medical records and how to mine this vast resource of medical information. Next, we will talk about systems biology, a relatively new scientific discipline that focuses on the challenges, tools, and methods of analyzing large biological systems, where the whole may be more than the sum of its molecular parts. In the last two units, we will talk about the effect of human genomic variation on human health and how this kind of knowledge is relevant in the fight against cancer. The course will include 8 additional units. Each unit will contain several recorded segments of conceptual discussion followed by practice sessions. In these sessions, participants will learn how to use the relevant tools we describe. There will be quizzes and assignments to make sure that the students understand and follow the lesson. So brace yourself and join us for this ride.